Compared to the vastness of space, humans may seem like a speck, yet even humans are comprised of even smaller units of matter. In this video, let's take a look at two specific organic molecules that shape who we are. All humans are different, physically and genetically. This is due to all of our different DNA, which is the instruction manuals for making proteins. In this specific situation, let's look at a protein that controls cell division. In turn, a protein that could be behind some cancers. The protein I am talking about is the Ras protein. Ras promotes cell division when activated. And when the protein is mutated, the Ras protein will remain activated. The constantly activated Ras protein means uncontrolled cell division, also known as cancer. Before we ask why the mutation is so important, let's first take a look at how a protein is made. The process starts at DNA. An enzyme called RNA polymerase travels along a section of DNA to create messaging RNA, or mRNA, a template for the protein. A ribosome then connects to the RNA. In the RNA, there are codons, a 3-letter block of nucleotides that are associated with a specific amino acid. The ribosome reads these codons and links a certain amino acid. A chain of amino acids is linked together in the ribosome, and it is the chain of acids that creates the protein. Proteins differ because of the different amino acids that each specific protein is comprised of. Different amino acids are made up of different elements, giving the proteins different properties. These contrasting structures then allow for different interactions of the proteins. Amino acids have the ability to form different IMFs, or intermolecular forces. This is the ability for one molecule to interact with another. Different IMFs include ionic bonds, London dispersion forces, or LDFs, and hydrogen bonding. These abilities allow amino acids to interact with each other. The different interactions in each amino acid are what shapes a specific protein. Regarding the Ras protein, there is a mutation in the DNA. This in turn creates a different functioning protein. A different amino acid is added instead of the intended one. In the non-mutated version of Ras, the amino acid glycine is the 11th amino acid added. However, in the mutated version, glycine is replaced with cysteine. The swap with amino acids changes the shape of the protein. The change in shape is due to the different properties the two amino acids have. Because the mutation favors the active state, there will be cancer caused. Restating, amino acids are linked together to create proteins. In order to create the bonds, amino acids must be able to chemically interact with each other. First, let's look at the structure of an amino acid. Every amino acid has the same basic structure. There is a central carbon bonded to an amine and a carboxyl. The center carbon also has a hydrogen and an R group. The R group is what makes each amino acid unique. The only factor that changes among each amino acid is the R group. Everything else remains the same. This uniform structure allows the amino acids to interact with each other. Since the body's pH is around 7, amino acids do not exist as seen. Instead, the amine is protonated and the carboxyl is deprotonated. If the amino acids attempted to link together, an acid-base reaction would occur. Therefore, in order for amino acids to bond together, there are special tools needed. In order to have the acids bond in the intended order, there must be a protectant on the amine and oxygen that are not intended to bond. Then you need a coupling agent to create a bond and overcome the acid-base reaction on the designated areas. Now, how are amino acids made? The body cannot actually produce their own amino acids, so our source of amino acids comes from the food we eat. Now, why are the R groups so important in defining an amino acid? Let's start with glycine. Glycine's R group is just another hydrogen. Hydrogen is not electronegative, and neither is carbon. Electronegativity is how strongly an element pulls electrons towards itself. This means that there are no polar charges in terms of the R group. The electrons are relatively evenly distributed. This means that the only interaction the R group can participate in is LDFs, London dispersion forces. An LDF is created at random. Since the electrons float around a molecule, there is a possibility for electrons to temporarily shift and create a temporary polar charge in order to interact with other molecules. This is a very weak IMF, as the charge is small and does not interact with charged or partially charged molecules. Cysteine, however, is another story. Cysteine's R group is a chain of carbon with 2 hydrogen, bonded with a sulfur and another hydrogen. In contrast to the hydrogen, sulfur is very electronegative, meaning that it pulls electrons towards itself through the bonds. This means the hydrogen electrons are being partially pulled away by the sulfur. The R group is polar because of this. The electron density is on the sulfur while the hydrogen has few electrons. This gives the hydrogen a big positive partial charge and the sulfur a big partial negative charge. These bigger and permanent charges allow for hydrogen bonding, which is the interaction between molecules with large permanent partial charges. These are strong IMFs and interact with other charged molecules. To recap, both glycine and cysteine are amino acids with the same basic structure. However, they both have different R groups. The 2 different R groups have different properties. Glycine's R group is an H and can only form weak LDF bonds between other nonpolar molecules. In contrast, cysteine's R group, CH2 with a sulfur and hydrogen, can form hydrogen bonds with other polar partially charged molecules. The different interactions the 2 amino acids can participate in means they interact differently within a protein. This changes the shape of the protein. In the case with the Ras protein, the changed shape is the reason why the protein is always activated and causing cancer. Who knows what this information can hold for the future?